Hello students, welcome to the course on Animal Diversity 1 Biology of Non-Chordates Unit 2. Let's learn the canal system in sponges. The instructor for this topic is Dr. E. Harini, Head Department of Zoology, St. Joseph's College for Women Autonomous, Vishakhapatnam. Contents: Learning Outcomes, Introduction, Types of Canal Systems, the asconoid, cycnoid and the leuconoid types, and the functions of the canal system. Learning outcomes. By the end of this lesson, students will be able to define the canal system in sponges and identify its main components like ostia, choanocytes, spongocoel, and osculum. Identify and describe the three main types of canal systems, the asconoid, cycnoid, and the leuconoid types. Explain how water currents move through the sponge body and their functional significance. Canal system in porifera. As we all know, all the sponges are marine and few are freshwater forms, but all of them have a distinguishing feature, that is the perforation of the body surface by numerous apertures or pores through which the water traverses the whole body and performs various physiological functions and is expelled out through the large opening called as the osculum, and collectively the pores, ostia, the osculum, they are together called as the canal system. And this system constitutes the most vital system in a sponge body because all the cell types of the sponges work centered around this system. Types of canal system in sponges. The arrangement and complexity of internal channels varies considerably in different sponges, and accordingly, the canal system has been divided into three types: the ascon type, the cycon type, and the leucon type. The asconoid type of canal system. This is the simplest form of canal system found in sponges. The example is Leucosolenia. As we all know that the body of sponges is pierced by a large number of minute openings known as ostia or incurrent pores, and these pores they extend from the epidermis to the spongocoel. And the spongocoel is a single large central cavity in the body of sponge, and this is lined by flagellated collar cells or choanocytes, and the spongocoel opens outside by a narrow opening known as the osculum. So here you can see the asconoid type of canal system, and you can see the course of water current moving through this. The outside seawater, the surrounding seawater enters through the ostia, which are the pores, and from there they move on to the spongocoel, and from the spongocoel they are sent out through the osculum to the exterior or to the outside. And here the flow of water is maintained by the beating of flagella of the collar cells, and the rate of water flow is slow because the large spongocoel already contains water which cannot be pushed out through the single osculum. The cycnoid type of canal system. The cycon type of canal system is characteristic of the cycnoid sponges, example Scypha and Grantia. And here the body wall is folded to form incurrent and radial canals which open into the central spongocoel by an opening called the apopyle. Both the canals are interconnected by minute pores called prosopiles, and the incurrent pores or ostia which are found on the outer surface of the body, they open into the incurrent canals which leads into the radial canal through minute openings called prosopiles, and these radial canals open into the central spongocoel by internal openings called as apopiles. In the next slide, you can see the diagram of the cycon type of canal system. So let's take a look at the cycon type of canal system. This is the cycon type of canal system, and when we take a closer look at the system, you can see the course of water current. Whatever water current is coming or is entering through the dermal ostia reaches the incurrent canals, and from the incurrent canals through the prosopiles they enter into the radial canal, and from the radial canal you can see that there are small openings called apopiles. So through these apopiles the water enters into the central spongocoel, and from the central spongocoel it is sent out through the osculum which is found at the free end over here. So this is the course of water current. The Leucnar type of canal system. In the Leucon type, the radial canals get divided into flagellated chambers by further folding of the body wall, and this type of canal system is characteristic of the spongilla. Here the radial symmetry is lost and therefore the canal system becomes very irregular. The flagellated chambers are small and lined by coinocytes, and all the other spaces are lined by pinacocytes. The incurrent canals open into the flagellated chambers through the prosopiles, and the flagellated chambers communicate with the excurrent canals through the apopiles. And these excurrent canals communicate with the outside through a small spongocele and an osculum. So you can see the picture in the next slide. So this is the image of the Leucnar type of canal system. You can see the different parts labeled: the ostia, the dermal ostia, incurrent chiles, prosopile, the flagellated chamber, the excurrent canals, the apopiles, and you can see the central spongocele and the osculum. Now water from the outside enters through the ostia, dermal ostia, and reach the incurrent canal. From the incurrent canal through the prosopiles, they enter into the flagellated chambers, and from the flagellated chamber through the apopiles, they enter into the excurrent canal. And from the excurrent canal, they move to the larger excurrent canal, and then they enter the small narrow spongocele, and from there through the osculum, the water is expelled outside. So this is the flow of water in the Leucnar type of canal system. The Leucnar type of canal system present three grades of evolution. They are the Euryphallus type, the apodal type, and the diplopodal type. So in the Euryphallus type, you can see that the flagellated chambers communicate directly by broad apertures called apopiles with the excurrent canals. Example is Placina. In the apodal type, the apopyle is drawn into a narrow canal called the aphodus, and this connects the flagellated chambers with the excurrent canal. Example, Geodia. In the diplodal type, some sponges, besides the aphodus, there is another tube called the prosodus, which is present between the incurrent canal and the flagellated chamber. Example, spongilla. We can see the images in the next slide. So here you can see the apodal type. So here in the apodal type, you can see that the apopyle is drawn into a narrow canal which is called the aphodus, and this is connected with the flagellated chamber. This is in the case of Geodia. In the diplodal type, along with the apodus, there is also another tube which is called as the prosodus, and this is called as the diplodal type. Moving on to the rhagon type. This is a type of canal system which is found in the Demospongiae. So this in turn arises by the direct rearrangement of the inner cell mass. So this rhagon type of sponge, it has a broad base and it is conical in shape with a single osculum at the tip or the summit. The basal wall is termed the hyposphere, which is devoid of any flagellated chambers, and the upper wall bearing a row of small oval flagellated chambers called as the spongosphere. So here the basal wall is not having any flagellated chambers, but the spongocele is bordered by oval flagellated chambers which are opening into it by these wide apopiles. And the dermal pores or the ostia, they open into the subdermal space and they get into the incurrent canals, and from the incurrent canals into the flagellated chambers, and these flagellated chambers have been formed by the breaking down of the radial canal, and the flagellated chambers are also lined with coinocytes which lead into the broad spongocele, and this spongocele opens to the outside through the osculum. So here you can see the course of water current which is moving from the ostia. It is moving through the subdermal space into the incurrent canals, from there through the prosopiles into the flagellated chambers, and from the flagellated chambers to the very broad opening apopiles to the excurrent canals, from there into the spongocele to the osculum and to the outside. So coming to the functions of the canal system helps the sponges in nutrition, respiration, excretion and reproduction. And the current of water which flows through the canal system brings the food and oxygen and takes away the carbon dioxide, the nitrogenous waste and the fecal matter. It carries the sperms from one sponge to another for fertilization of the ova. And the canal system increases the surface area of sponges and increases the volume. That is why the ratio of surface area and the volume remain fixed. So let's summarize. The canal system in sponges is a unique network of pores and channels that facilitates the continuous flow of water through the sponge's body, enabling essential physiological functions such as feeding, respiration, and excretion. And water enters through the numerous minute openings called ostia. They pass through a series of canals and chambers lined with flagellated collar cells that trap the food particle and exits through a large opening known as the osculum. And depending upon the structural complexity, the sponges have been classified into three main types of the canal system: the ascon type, the cycon type, and the leucon type, progressing from simple to highly efficient designs. So this system is very unique for the poriferans and it forms the functional core of the sponge biology, ensuring survival through effective water filtration and nutrient absorption. So these are the references for the topic discussed. Thank you for your patient listening.